Thanks everybody for joining me again. This time I have, I think, a solid internet connection, and I'm excited to tell you about our first results from the OpenKnot challenge. This began last fall in 2022. It's totally new for Eterna since the last eternacon, and there has been very rapid progress in just a few months. So start with a bit of the motivation here. So it's been a longstanding goal of RNA science and medicine to be able to discover and to design complex RNA structures. We just got, a really beautiful overview of these structures from Quentin and from Joe and others, and we'll hear more about this throughout eternacon. Many of us in the field started to jump into discovery of RNA structures when the COVID pandemic began. And I'm showing you here a figure from a paper that I was involved in, as well as Joe Townley, where we scanned coronavirus genomes to look for parts of that RNA that might form interesting, complicated structures, which could then become targets for designing small molecule drugs, pills that you could take to fight the viral infection. And while the field of while we still don't have FDA-approved medicines that target these RNAs against viruses, there are existing antivirals and antibacterials that have been approved. And actually, many of you have probably taken something called Augmentin, which contains variants of erythromycin, which is an which targets the ribosome at the RNA, and it's actually the second antibiotic ever discovered by humans. And variants of that are still in the clinic, and you can get them to treat your infections. Okay, so there's a lot of effort on trying to design those drugs against these kinds of targets, but a big stumbling block that a lot of people are facing is just if you have a new virus, if you have a gene that you think is important for Alzheimer's or for cancer, then what parts of that RNA gene actually fold into structures that you can target. So these are example secondary and tertiary structures that we pulled together using actually chemical mapping data in the literature and in our own lab, including our Eterna earlier on structural labs, and to propose some of these models. But what if you could do this what if you could do this ahead of time for every potentially pandemic human virus? And what if you could do it without using any chemical mapping? What if you could just predict the full complicated three-dimensional structure of an RNA without having to do the crystallography or NMR that Quentin and Joe told us about? So that's been a long-standing dream of mine and of the field in general. I was fortunate to be part of a competition last year called the CASP competition, which is the worldwide challenge. It's been going on every 2 years for the last at least 20 years, almost 30 years now. It's a protein structure prediction contest. If any of you have heard of the AlphaFold2 AI method, that's largely solved the problem of protein structure prediction. It was elicited because this challenge goes on every 2 years and rigorously ranks computational and other predictive methods for protein structure. So I helped organize and assess a RNA structure prediction exercise as for the first time in CASP last year. And we were quite delighted that out of several structures where our lab and collaborators and other folks in the field, we had solved experimental structures of these RNAs using crystallography and cryo-EM. And then what I'm showing you here is those structures shown in green, and blue are blind predictions that were made by different groups. So this is great to see that the field at large can make these kinds of predictions. And these are actually RNAs. This one down here was in Quentin's talk. Actually, it was the one that he put in front of Tony Fauci's slide as hopefully a kind of nanostructure we'll see in medicine soon. But it turns out none of the methods that were used to predict these structures, although about at least 15 AlphaFold-like methods were submitted in the CASP competitions, none of them produced models that were as accurate as what basically human experts could do using their own intuition and often using kind of semi-automated computational tools that they had developed in their own labs to supplement their intuition. So the deep learning revolution hasn't hit RNA, at least RNA three-dimensional structure yet. But if it did, then you could imagine scanning viral genomes or genes for Alzheimer's and finding these elaborate structures. And you could also imagine designing nanomedicines like new mRNA therapeutics that would have these structures. Okay, so that's the motivation. What would it take to get to, let's say, something like an AlphaFold for RNA? Well, for any deep learning approach to machine learning, you need three ingredients. You need some kind of model that can be trained, usually a deep neural network. You need some kind of way to evaluate whether that neural network is producing like real predictions. And then you need data for training, typically a lot of data. And so I've just actually described to you that there were about 15 AlphaFold-like models that were submitted in CASP for RNA 3D prediction. So those exist, and many of those are in open source repositories. So those already exist. And now we have rigorous evaluation in the form of things like the CASP trials, as well as a competition that's been going on for longer called the RNA Puzzles competition, which runs not just every 2 years, but is a running competition. So we have two forums for evaluation. But what seems to be missing, I think everyone in this field has hypothesized that we're missing data, sufficient data to train an artificial neural network. And so then you can say, well, could we just apply the experimental methods that we have developed as a field to millions of RNAs at once? And actually, there's this funny chicken and egg question where I actually don't have a list of 1 million RNAs that might have diverse and interesting RNA structures to begin with. We also don't actually have experimental methods that have the throughput to go and get the coordinates of the RNA three-dimensional structures that such molecules would form. But there's an even more basic problem in that we don't have a list of those RNAs. So there are databases of proposed structured RNAs. They have names like RFAM, but they only contain on the order of a few thousand families. And and for most of those families, there aren't more than, let's say, a handful of really distinct sequences. So we have this really major gap. And so, and just to also kind of back up that idea that you need millions of images or millions of data, this is a figure from a popular science article in the Financial Times that came out earlier this year, where they pointed out that the deep learning revolution began a little more than a decade ago with an image classification contest called the ImageNet contest. And this data set involved about 15 million images. So that's when things started to really get cooking for deep learning. And prior data sets showed some possible, proofs of concept that deep learning would work, but this is really the data set and the task that pushed deep learning into prominence. Of course, nowadays, current AIs are trained on something like billions of images or billions of sentences of text, but it starts around the scale of 1 million or a few million. Okay, so how do we get there? So the idea that that we've been exploring largely on Eterna is to crowdsource the discovery of ideally millions of interesting folded RNA sequences. And there are other sources. I've mentioned RFAM, which is essentially also a crowdsourced database of potential structures, crowdsourced to the expert RNA community. There's also just stuff that's in nature. So you could imagine looking at every segment of a coronavirus genome or other kinds of viral genomes. But this looks like the perfect kind of problem for Eterna. Eterna's special sauce, I feel, is going into a problem where there's low data and then discovering, having this community of humans talk to each other, go through every paper, go through every database, and then think and inspect and say, here are the RNAs that we might fold into structures. And Jeff's talk right before this was a spectacular example of that kind of inspection, now with the superpower of new tools, the OpenKnot tool, to go and find those structures. And the idea is maybe we could start with a round where we try to get thousands of these RNAs, and then maybe 10,000, then 100,000, but start small, but then start scaling up till we get to a large data set. And the way to tell whether an RNA has a complex structure is through an approach called chemical mapping, which you've heard a bit about, the DMS mapping approach. You've heard about that from Joe Yesselman already, and I'll in a second give you a brief recap of how that works. But for each of the, but it basically means that for any RNA you make up to a length of about 100, and we can now push this up to 200, 300, you can get data on which parts of the RNA are forming structure versus not, and then that allows you to test whether it formed the structure you think that it folded up into. And then from there, even with 1,000 of these, you can go and rank different methods that allow you to model or design RNA structure. Those could be our community's intuition. Those could be automated methods like NUPAC, like what we've heard about, or EternaFold, or even existing neural nets, like Spot RNA, which was developed for RNA secondary structure prediction and pseudoknot prediction about three or 4 years ago, or the deep learning networks that came out of our own work in the open vaccine competition, where we collaborated with Kaggle and elicited about 1,600 teams took part in the Kaggle competition and produced mainly deep learning based approaches to model our mRNA degradation data. So imagine you had these data, but our complex RNA structures, you start ranking these models or refining them, then we can use these models to then get the next set of structures either automatically or even better. We can use those tools ourselves as humans to go curate a bigger and bigger data set. So that was the idea. And hopefully after enough rounds of this, we get to the point where we have models that themselves, without our help, can go and discover and design complex RNA structures. Okay, that's the hope. And we have now gone through, if you count the pilot round, we've gone through 3 rounds of what we call the OpenKnot challenge. We decided early on to focus, well, although we're interested in any kind of RNA that forms complicated structure, including the tetraloop receptors that you heard about in Joe's talk and in Quentin's talk, we decided that we could bootstrap our way to those more interesting structures by taking advantage of previous algorithms that produced models of arguably the simplest kind of tertiary structure or complex structure, which are pseudoknots. And there's about a dozen of these pseudoknot folding algorithms already in the literature, whereas there are very few that model 3-dimensional structure. So, so we decided to focus on knots, pseudoknots, called this OpenKnot challenge. And as many of you know, we have open right now on Eterna, the OpenKnot round 3, and where you can type in any 130 mer RNA you want. It doesn't actually have to form a pseudoknot if you don't want, but you can go in there and put in basically any sequence you want, and we'll synthesize it and chemically map it for you. Okay, so I want to show you results from the very first pilot round. So before doing anything, actually even before developing the high throughput experimental protocol, I asked the Eterna community back in, I guess, October, November, can you just make a bunch of RNAs you think will form pseudoknots? And the response was fantastic. We had, I think, 40 to 50 players come and play these very, very difficult labs. The folding engine is very slow. It relies on a version of an algorithm called NUPAC using a pseudoknot folding flag that we happen to have ready to go in Eterna, but it's very, very slow. And what I'm showing you here is the sequences are plotted based on, and they're far away from each other or close to each other based on how similar their sequences are. This is the so-called t-SNE plot. But suffice to say, these are colored by all the different players who contributed, and there's a huge amount of diversity. Okay, that was awesome to see. And I'll tell you about what some of these are in a second. Once we had these sequences, that motivated us to go develop protocols to go and carry out chemical mapping of all of these RNAs. And so I'm not going to dive into the details here, but I'll give you a brief overview. If you want a masterly presentation of how we do chemical mapping, Ray Huang from my lab gave a very concise but thorough description of our protocol in the town hall from this Monday. And if you log into the Eterna forums, you'll be able to get a link in one of our forum threads that Jonathan posted. But basically, there are now companies where if you send them actually 1 million sequences, we literally just did this yesterday. They a week or two later will send you a bag of test tube where they've synthesized all of those sequences. as DNA. And then there's not very much of each of those DNA sequences, but our lab has revived a technique from the early 90s called emulsion PCR that allow us to amplify up. So we have lots and lots of DNA that corresponds to each of your designs. And then all in one test tube in a big pool, we put in a machine called RNA polymerase, a little tiny nanomachine that then transcribes from the DNA to produce all the RNA molecules, okay? And lots of copies of each of your RNA molecules. Then we take advantage of chemical modifiers that mark out the bases that are not forming pseudoknots, that are not inside the pairs. And those mark these open or loop bases. And then we look for protections later on of the parts where you have tried to design your RNAs to form base pairs. And these chemical modifiers have been developed in several labs, not my lab, but we've adapted modifiers from Kevin Weeks's lab and from Danny Incarnato and Tony Musto's work and their protocols to then map these RNAs. And then it turns out then to figure out where the marks are, you have to convert this back to DNA. That lets us take advantage of extremely cheap DNA sequencing methods that are out there. And to do that RNA to DNA conversion, there's another step called reverse transcriptase. Then there's another innovation that Ray's come up with that allows us to get very pure and pristine data from purified libraries. I'm not going to skip that step. And then we send off these final DNA libraries that harbor information about where your RNAs were structured or unstructured. We send that off to another company. Okay, and it turns out through the innovations that Ray has largely put together, including this purification step, as well as these innovations we've adapted from other labs to get very strong chemical mapping signals. It turns out that when we started this project back in October, November, I was worried that we wouldn't be able to get more than about 10,000 data on about 10,000 molecules. But we started anyway. And then it looks like now, if things go well, we're crossing our fingers, we'll be able to get data on up to 1 million molecules per experimental round. Okay, so what do we do with those data? Well, we got those data for the pilot round a couple of months ago, and we released them on Eterna. I thought I'd just quickly explain how we scored them, and Jeff actually described some of these scores. They go from 0 to 100. How do I identify RNAs that actually form these interesting structures? The way that we do it is I'm showing the experimental data here on the top line, and red means that there's a segment of the RNA that was unpaired and reactive. And the parts that are white or very light red or light blue here are parts that were not very reactive. So they're likely forming some kind of pairing. And typically, we weren't sure going in whether the NUPAC folding engine does a good job of actually predicting the structure that you're designing for. So what we've decided to do is, this is work from Rachel Kresh and Grace Nye and Thomas Karajanes. We've actually run basically every algorithm that we can find that tries to predict a secondary structure. Some of these take minutes, like tens of minutes to run on your RNA sequence. So we run this on a supercomputer. And we check and see then, do any, and if so, which ones of these sequences do they match the experimental mapping pattern, okay? And we use a classic Eterna score, which goes from 0 to 100, and that's what we use to rank these structures. Sometimes many of these different packages independently produce basically the same structure, okay? And don't be too fooled by the square brackets here changing to parentheses. Some of you aficionados out there will recognize that we're showing actually the same structure over and over again here. And we take the predictions of all algorithms, the best match algorithm, and everything that has an Eterna score within 5 units of it. And we say, well, those are all probably good shots on target for predicting the dominant structure that the RNA forms. And then for those, so that's this set over here. And for this set, we then go look, well, out of those predictions, which of the pairings here actually cross each other to form pseudoknots? And those are the ones that are boxed here in blue. And then we go look back at the raw data and say, well, for those segments, are they actually protected from chemical probing? In this case, actually, they all are. And so this design it ends up with a good score. It's going to recur in a later slide. And the final score that we give out to players here is the Eterna score for the best design, the best fit the best fit structures for the data for your design. And then that's half the score. And the other half of the score is how protected are the pairs that are crossing or that actually form a pseudoknot. Okay, so this final score goes from 0 to 100. And if you want a more detail, there's some, it's averaged over all the models that appear to be within reasonable fits or within 5 of the best fit. And there's some other fine details. And if you want to understand more of the details or even want to see the code, it's available publicly in this GitHub repository, along with a README that explains how it's calculated. And if you plot what these scores are, excuse me, from our pilot round, you'll find that there's a peak of the OpenKnot scores. Fortunately, no one did much worse than 50. All of the RNAs you made matched to some kind of structure. And so that's these designs here. But there's a peak. There's actually a set of structures that are separated from the other ones out here around 85. And those, I'll show you a few of these, but I looked at basically all of these by eye, and they all look to me like they're reasonable, like there aren't any weirdo artifacts that pop up here. And they look like they really could be forming pseudoknots. They're ones that we would love to follow up on, and we are following up on through independent experimental methods. Okay, so what are some of these top hits? Oh, I also want to point out this. I'm coloring again that plot from before where we spread out all the designs by sequence. And this is also cool to see. It wasn't just that those 10% of the designs were all coming from, from Riv or from Digital Embrace. There are some clusters of designs like this one, but there's designs from essentially everyone on Eterna, and they're spread out throughout sequence space. So this is also really important going forward. We don't want to just have the same pseudoknot showing up over and over again. We really want to have a lot of examples of pseudoknots that folded properly, as well as ones that don't. Okay. So let's take a look at some of these top hits, because they're super interesting. I mean, and I could spend hours just talking about every single one of these designs and asking all of you how you came up with them. But I thought I'd just go through some of the ones that are on the leaderboard from this pilot round. The top two were both from Jill or Digital Embrace. And Jill, please correct me if I'm wrong, but these came from actually a cloud lab round from, I think, 8 or 9 years ago. That was originally created by Cody Geary, who some of you may know is one of the co-inventors of the RNA origami technology and is a co-author on some of the papers on nanostructures and co-creator of some of the molecules that Quentin actually showed in his talk. So Cody is someone who has known Eterna for a long time, and I've known Cody since for a long time as well. So he, along with Ebby Anderson in Denmark, created a lab where they created this little tiny RNA, double-stranded RNA origami. And I think Jill then put in a couple of the winners from that old lab and asked, do they actually form, this is the pseudoknot here between the blue and the green. That's the kissing loop that produces this complex structure. And indeed, these are here in actually they look like the best designs that we have from the pilot, at least. And then several of the designs, this is actually reassuring to see. Although we love having a diverse set of designs, it also feels good when you see kind of if independent players put in a very similar designs and they all get ranked together at the top. That helps us verify the robustness of the experimental procedure. And that was really cool to see here. So it turned out that many of you out there dug into existing databases proposing pseudoknots in, for example, viral structures or synthetic RNA nanostructures. PKB is one of them, pseudo-based. And it turns out the pseudo-base for PKB 131, 132, and 133, one of them I actually showed in the previous slide, produced several of the winners, 1, 2, 3, 4, at least 5 of the winners. And I guess we can ask Riv if her, I think some of Riv's designs also were drawn from PKB. I wasn't sure if this one was. But what are PKB 131 to 133? Well, it turns out these are aptamers that were discovered through in vitro selection, I think in the 1990s, that bind to a protein called human nerve growth factor. And there's no 3-dimensional structure solved experimentally of this RNA, but we have pretty good evidence that this thing is forming an interesting pseudoknot in solution. And then last, I think this is most exciting for me, are designs that were totally synthetic. As far as I can tell, these have no relation to anything in nature. Okay, so we want these. In fact, we want lots of these because probably at some point we're going to saturate all the pseudoknots we know about in nature. So I'm going to highlight a couple here. One is from SP Vincent, and actually this was in one of the examples in J. Anderson Lee's talk. F65, and I just grabbed a screenshot from Eterna. I guess I should have grabbed an ArcKnot plot too. That would have been nice. And then a couple of spots below, basically the same score, is Jesu 32 from Jester. Totally different fold. Okay, so and as far as I can tell, these are not related to anything in nature. Although I hope that both of you, as well as other players, will correct me if I'm saying the wrong thing. I also want to highlight here that something that I mentioned earlier this morning. SP Vincent has been with Eterna for a very long time. Actually, I guess before its launch, and it's really great to have you here again, here at eternacon. And so we have, players who I'd consider the really core Eterna community. And then we also have players like Jester who I think must have joined around the same time that we launched these puzzles, and then has submitted so many beautiful different designs. And I'm not sure if Jester is here. I don't know what everyone's real life name is compared to their login names, but I wanted to thank you as well as everyone else who's contributed to these labs. And particularly excited about some of these more synthetic sequences that are now coming in. Okay, so there's a bunch of other stuff that's happened. Some of this is documented in the labs themselves in the conclusions page, which I've filled out. But I thought Jeff has already noted that it looks like none of the algorithms that we have in Eterna are particularly great at predicting whether something has a pseudoknot. And actually, we've gone through all of the algorithms that we have and compare them to our data. One way to rank the algorithms is based on their correlation coefficient of their predictions, whether something's paired or unpaired, to the actual chemical mapping data we have. One of the things that we see is actually NUPAC does not do very well in these. Actually, it's kind of down at the bottom of the list here. And I'm showing you here a list that look at just the pseudoknot 50 and pseudoknot 90 different pilot rounds. So you can get a sense for how robust these rankings are. But Eterna NUPAC is usually near the bottom of the list. Some of the neural networks that exist, spot RNA and E2E fold, are just terrible. They're really the worst, like by a lot. But it looks like the algorithms that produce structural guesses that best match our data are EternaFold, and then which doesn't predict pseudoknots, and then something called EternaFold Hungarian, and also maybe EternaFold ThreshKnot. EternaFold ThreshKnot, actually, these kinds of results are what prompted us to then make EternaFold ThreshKnot the folding engine for Eterna. So Thomas put that in for the current round. And EternaFold Hungarian is actually slightly better. We didn't realize this at the time, but we've gotten more data since then. And this is a new algorithm, semi-new algorithm developed by Rachel Kresh in my lab. She's taking EternaFold base pair probability output, kind of like what Jeff was showing, and then she has a different kind of heuristic for figuring out how to get a predicted pseudoknot. So these are interesting, and maybe these will become folding engines for Eterna going forward. I should give one caveat here, that it looks like these engines, actually out of the designs that really appear to form pseudoknots that have good high OpenKnot scores above 80, that it turns out these engines are, they don't predict them to be, most of them are not predicted to be pseudoknots within these top engines. So there's this funny thing where you actually might want to use a different engine for detecting pseudoknots than you might want to use for just predicting these data in general. And so that is something that we're still figuring out. I will know the answer in the next month or two. It'll almost certainly influence how we're setting up future rounds of this Eterna puzzle. But I think it's super exciting because these are the first turns of the crank in that strategy where we're using our data, especially these diverse designs from the Eterna community, to then go and figure out actually what algorithms can you use to go and design and discover RNAs. Okay, so it's happening. Okay, so I thought I'd end here by announcing some of the upcoming events within this particular challenge. So in August, our goal is to have the data for OpenKnot round 1. That's about 120,000 sequences, which they're getting, actually the data are getting sequenced right now in Northern California. And then we're crossing our fingers that the data are good quality. And if that's true, then we'll be able to release those data to you by August. We hope to get these to you by, and we also will hopefully have data for a special, a highly specialized lab challenge called the Pseudoknot Detective Challenge, where we asked you as a community to explicitly find pseudoknots that you knew that you saw them in the PDB or pseudo base with longer lengths. These are more expensive for us to make, but we wanted a good training set where they're assured to have interesting three-dimensional structures. So we hope to have those data collected as well in August. And in fact, we hope to have these available to you as a community so that they can inform what you're putting together for OpenKnot round 3, which is ongoing and which will complete on August 15th. Okay, so that's August. In September, we hope to have the data for OpenKnot round 2 made widely available, as well as some supplementary designs that our lab is putting together. These actually include some designs from the RNA make software that Joe Yesselman invented, which Jill asked him about. And thank you, Joe, for your help in helping us get some designs from there with tetraloop receptors, as well as other RNAs that we've scraped from other databases, expert databases. But of course, the designs I'm most excited about are the ones that have come in from Eterna. Around that point of September, OpenKnot will still continue while we're taking a look at these data and feeding back from these data. Although it seems like to get the most diversity now, it's pretty amazing that Eternal players are now scanning entire genomes as well as mRNAs from humans to go look for interesting structures already. We also are going to start focusing efforts on a subset of structures to help ensure that we're all talking about the same structures and trying to figure out principles for getting these RNAs to fold up. So we currently have about a 10% hit rate on finding RNAs that have pseudoknots. But why can't we get it so that every time we see an interesting pseudoknot structure, can we design a sequence that folds up into it? So I think that's something that we can promote by having these 10 structures a month or so. And current plan is this is TBD, but we're planning to synthesize roughly quarterly, so every 3 months, all the designs that are coming in. And the structures that we'll look at per month ideally should be coming from the community. We've seeded in the current lab about 10 structures where Jill has talked to players as well as Rachel, my lab, for some fun structures to start with. But ideally, these will be coming broadly from the community. They can come from your previous submissions from the OpenKnot rounds. And as of, I think this morning or late last night, we have set the puzzle maker, those of you who have access to the puzzle maker, you can now create your own pseudoknot puzzles with Eterna fold thresh knots as the folding engine. And so we'll be looking at those as well for suggestions for what we can use for these upcoming rounds. Okay. And then in October, the machine learning efforts to take these hundreds of thousands and maybe a million sequences, those will begin. Those are kind of beginning. We're preparing that now. We are in contact with several experts across the machine learning community. And there we'll be trying to train something. I mean, tentatively calling it EternaNet, but I don't know what we're going to end up calling it. The goal there will be to take previous data and then to predict the data for OpenKnot round 3, which we will have collected in August. But our plan is actually to wait to synthesize those RNAs and chemically map them until after these predictions are made. That is the most rigorous way to do the challenge. And then in December, we'll have the data and we'll figure out whether these methods are any better. And then that puts us into early 2024, where we'll be hopefully releasing an updated folding algorithm onto Eterna. Maybe we'll have some preliminary stuff before that, but I think this is going to be the big event. If it's not a particularly great algorithm still, we'll iterate. Okay, we'll get some more designs based on this new algorithm and then try to figure out how to break it, how to find out where it's going wrong. Also, if it's any good, we'll also hopefully automatically rescan all of viruses, bacteria, parasites, human genomes. If the Eterna community doesn't do that already, then we'll just go ahead and do that automatically on a supercomputer. Hopefully it'll be fast enough. Okay, so that's the overall schedule. I want you to think also beyond that. So this is the first time, so on Eterna, as I mentioned earlier this morning, we've repeatedly gone into these problems where we have very few data. We really need human intuition. We ideally need a community of experts who collaborate to solve hard problems with few data. And what we're piloting this year with OpenKnot is actually starting a project on Eterna that eventually goes up to a scale of 1 million or more sequences, and will hopefully then allow us to see deep learning approaches. But I think this is going to be extremely powerful going forward. This idea of let's collectively solve problems on Eterna, and then publish how we solve them, the designs and the data, and then that's the handoff to machine learning methods. Then we go into harder and harder problems. So the question for the community to start thinking about is, what are those harder and harder problems? So one thing that we haven't done much of in Eterna so far is to look at RNA structure within cells. So RNAs interact with all sorts of other machines, including proteins inside cells, and it's like totally unknown how much RNA folding is really perturbed. Can you get novel RNA structures inside cells? Can you, and for things like mRNA vaccines and therapeutics, you have to get the RNAs to form their structures inside cells. So that's potentially a future. We've heard earlier today about aptamers. These usually come from in vitro evolution or selection experiments, but what if we could figure out as a community, you give me some target protein or small molecule, can we design an RNA that folds up in 3 dimensions and then senses that molecule? I think that would definitely revolutionize all of biotechnology and potentially a lot of biology, and it would require a really deep understanding of how RNAs fold in 3 dimensions. And then I want to highlight, this has sort of been, for me personally, something that I want to do for, I guess since I was a student. Can we design not just RNAs that have particular structures or that bind other molecules, can we design RNAs that are actually like machines that go and sense other various inputs, and then chug through a few different states and then create some non-trivial output. So I mentioned earlier this morning the idea of making computers for smart medicines. If we could figure out principles that would allow us to process more than 4, more than 8, ideally up to 16 inputs, and then produce a bunch of outputs, that would be incredible. And I feel like we actually kind of have the beginning tools to do this. We have been working on ribosomes to make new materials. So far, we've been able to figure out how to stabilize some of these ribosomes, but can we actually just design a sequence that produces a polyketide antibiotic rather than a protein? And then finally, there's RNAs. There's a holy grail problem. Many scientists believe that at an early stage of life, there was just RNA, and RNA figured out a way to replicate itself, and that was a key event in the origin of life. But currently, there's still not quite a proof of concept of a molecule that can do that. So being able to make such a little machine and then understand what it would take to evolve that, I think will shed a lot of light into potentially how life began on Earth, but also how we could potentially further improve biology, and also how to avoid self-replicating parasites like viruses. So this is, I think, a very potentially interesting front. All of these require understanding and getting high throughput data sets and designing stuff that moves, that really has actual motion versus time. And so there's a lot of challenges to develop the experiments to go and test the designs, to create the game mechanics so that you can actually interactively design RNAs that have a shot of having these functions. And then eventually creating automated methods based on designs. But these feel like really, really deep and interesting problems that would change the world if we could solve them. So with that, I just want to kind of provoke ideas and discussion. I want to thank the folks who have been working on the OpenKnot challenge. These are folks in my lab. Ray Huang is here in the front, who I've mentioned, experimentalist. Grace Nye in the back has given talks at town halls on the viral scans. Rachel Kresh I've also mentioned. And so she, for the last week, has been putting together this library that includes OpenKnot round 2 for us to synthesize. Eterna devs, including everyone in the call. I've already mentioned folks who have been helping us on the experimental side of things. These are some machine learning folks who I've been reaching out to, computer scientists mainly. And then everyone here who's been contributing to the pilot rounds. This is a list of folks who contributed, I think, to PK50. I think many of you are actually here on the call. So thank you so much for contributing, and hope to see you in OpenKnot round 3 and beyond. And then just want to end here. For what to do next, any of you who are still interested in OpenKnot, the puzzle maker, as of this morning, will let you make puzzles that where you can challenge each other to make to fold into crazy pseudoknots. So I'm looking forward to seeing what you come up with. OpenKnot round 3 is open. So let's make find as many RNAs that we think might fold into complicated pseudoknots as you can. Right now, there's a really beautiful set of designs coming from largely from human mRNAs, that's very exciting. But also think about other sources of complicated complex RNA structures. And then fairly soon, we'll announce this on a news item. We'll be making the data that we have on Eterna more easily accessible to the broader scientific community through the RNA mapping database. So at that point, please, those of you who are interested in machine learning or AI, we hope that you will take a look at those data and get your friends involved and your kids and your nephews and to come and start developing these AI approaches. Okay, I'll stop there.